Hello, everyone. My name is Tucker Hallmark, and I'm the manufacturing manager of lipidomics at Polar Lipids. It's my pleasure to welcome you all to the second webinar in our Explore the Lipidome series. And I'm excited to introduce today's speaker, Dr. Federico Torta. Dr. Torta is a research assistant professor at the Singapore Lipidomics Incubator, SLING, which is part of the Department of Biochemistry and Precision Medicine Translational Research Program at the National University of Singapore. He's also co-director of the Lipidomics Unit at SLING-MAS, the Singapore National Laboratory for Mass Spectrometry. He received his PhD in biochemistry from the University of Parma in Italy, and his expertise includes sphingolipidomics, high-throughput technologies for the assessment of lipidomes in large human cohorts, and the study of protein lipid complexes. His talk today is entitled Harmonization in Biological Reference Ranges in Lipidomics. And if you have any questions during the talk, please insert them into the Q&A box, and we will take questions at the end. And with that, I will hand over to Federico.
Thank you, Tucker. I'll try to share my screen now. I think now you can see my slides in presentation mode. Is that all right, Tucker? Can I continue?
Yes. Yeah. Perfect.
Okay, perfect. Okay, thank you very much, Tucker, for the introduction and for giving me the opportunity to present all about our efforts towards harmonization in lipidomics and how we would like to measure biological reference ranges of lipids in human blood samples. Now, why we want to do that? Okay. We want to do that because dysregulation of lipid metabolism has been associated with many diseases, such as cardiovascular diseases, diabetes, and also many others. And lipidomics has given a lot of useful information to try to decipher the mechanisms behind these diseases. And one of the big promises of lipidomics was to finally be translated into clinical diagnostics and also to allow precision medicine approaches in which we could more precisely characterize a disease and its phenotype. Although there are still limitations to these approaches, and probably one of the reasons for why the biomarkers that has been generated by lipidomic approach didn't manage to reach the clinical environment is probably because our methodologies are still poorly performing in terms of quantitation, reproducibility, and comparability. But this is what the clinicians would need, because the clinicians base their diagnosis on reference values. And to get these reference values, we need methodologies that are performing under certain requirements in terms of quantitation and reproducibility. Now, the first call for harmonization in lipidomics was done by John Bowden and then published this paper in 2017. So John sent around the world to 35 different labs a representative plasma sample. And this case was from the Nist, so it was the standard reference material 1950 plasma. And then asked to all the participants to measure as many lipids as they could with their preferred method. So from the results of publishing this paper, we saw that there are different preferences between labs. Many labs prefer targeted versus untargeted lipidomics. Others prefer to use LC versus direct infusion, and others, again, prefer to use high-resolution mass spec compared to low-resolution one. But another thing that became clear after looking at the final results published in the paper was that there was a huge disparity in terms of concentration levels reported by the different labs. Indeed, let me see if I can have a pointer. Indeed, for example, in the case of this specific PC species, if we look at the dispersion of the values reported by the different labs, is very high. And even if we consider the consensus value that was calculated using the measurement given by all the labs, almost no lab measured the value that we consider as a consensus estimate. So this was not, this paper was not intended as a collection of reference value, but it gave us an idea of the variability that existed in our community, and that was very important. Now, where is all this variability coming from? It comes from many different steps of the experiment. It can come from different extraction techniques, from different type of internal standards used, from the different introduction method into the mass spec, and of course also from the different analyzers used. Now, what we did here, and Alex, who was a former member of our lab, did, was to start simple and then look at the influence of the sample introduction methods on the concentration level that we measured. So we kept the experiment quite simple. We used the same mass spec for all our measurement. In this case, we chose to use a thermal Q exactive. And then we connected either with a direct infusion device or with an LC. And when we use the LC, we compared a reverse phase separation versus a helix separation. The experiment was the following, also very simple. We took two different commercial plasma samples. One we call it as our long-term reference plasma, and the other one was the Nist SRM 1950. Then we added the internal standard to the plasma, one internal standard per class in this case. We performed the same lipid extraction, and then we did different aliquots, one for direct infusion, one for LC helix, and one for LC reverse phase. And then we calculated the concentration of each lipid measured, and we divided the R of the peak by the R of the internal standard, and then multiplied by the concentration of the internal standards. And we obtained the concentration of the same lipid with the three different methods. So these are the results. After filtering for our QC criteria that you can see here, we got a certain total number of lipids quantified by each method. And not surprisingly, we found out that we had more species quantified in reverse phase and less with direct infusion. And of course, this is due to the fact that when we do a reverse phase separation, we increase the sensitivity of the method. Then between all these lipids, we selected the 90 lipids that were measured in all the three methods. And then we further decreased this number to the between these 90 to the ones for which a consensus value concentration was reported in Bowden's paper. Then what we did, we analyzed the lipid levels by PCA. And what we saw was the following. So we clearly could separate the two different plasma samples because the lipid profile is different. So the LTR was separated clearly by the Nist sample by all the three methods, fusion, reverse phase, and healing. But unfortunately, there was also a very clear difference between the lipid levels measured by the three different methods. So you see here there's very good separation between the three different methods. And indeed, if we represent our results in this way, so these are the lipid species that were in common between the three different methods, and these are the concentration measured by the three different methods. You see that some of the species were found with quite actually very similar concentration values, and this is the case of LPC and some of the PCs. But if we look at polyunsaturated PC species, or for example, TG is even clearer, the difference was quite big. And indeed, if we try to quantify this difference in terms of RSD percentage between the three different methods, you see that most of the lipid species showed an RSD percentage between 40 and 80%, which means that there was a big variability between the three different methods. But also what I would like you to notice is how low is the variability for these three lipid species. Now, these three lipid species in the green box here are the ones for which we added a species-specific internal standard, which means it was the same lipid but deuterated. So this also tells us that only species-specific but not class-specific standards correct for ionization differences in this case. Now, after seeing all these differences, we thought, how can we convert the MS signal to a more meaningful value that is more reproducible and more comparable? And the idea was to use calibration with external standard reference, which is a procedure that is well known in clinical chemistry, for example. So now, how to choose and use a reference material to perform this external calibration? So the reference material has to be reflective of the experimental metrics. So in this case, we are using plasma. We have to use a reference material, which is plasma. It has to be interspersed among the study samples and not mixed with the study samples. So here you see an example of how you could design your batch. You have blank, you have QCs, and then you have all your study samples, and then you interspersed one reference material sample per batch. And then the reference material sample has to be prepared alongside the study samples in each batch. So they have to be prepared together. There are different types of reference material. In this case, we recommended the use of the SRM 1950 because it's easily available and has been characterized quite well by our community. But of course, there are also other materials that either the Nist or other companies are working on. And actually, if you're interested to know a bit more about reference materials in metabolomics and lipidomics, this paper has just been published by the Metabolomics Quality Assurance and Quality Control Consortium. So you can read this paper for more information. Now, how do we use this material? So The idea is to perform a single point calibration. So we will measure the concentration of each lipid in our sample, and we will measure the concentration of the same lipid in our reference material. And then we will calculate a ratio between these two values so that we will generate a normalized concentration value, or as it is, a relative concentration value relative to the reference material. Now let's see what happens when we apply this idea. So these are data in the PCA that I just showed, pre-normalization, where the two plasma lipids are separated, but also the three different methods are separated. Now, when I apply this correction factor and I replot the same data after normalization, I now get this type of PCA in which the LTR and the Nist plasma sample are still well separated, which is good because they have different lipid profiles. But in this case, for each of the two plasma samples, the three different introduction methods gave the same results. They are very, very similar results. So the conclusion is that in this case, the normalization to the reference material can correct for method-dependent systematic differences. And indeed, if we plot again the data by lipid species like we did before, now we see that in this case, if we compare the concentration obtained with the three different methods, the levels are absolutely the same. And again, if we calculate the RSD between the different methods, the orange bars represent the data of RSD percentage between the three methods before normalization, and the green one are after normalization. You see that after applying this normalization, all the RSD values are below 10%, again telling us that the three different methods gave the same result. Now, what is the practical relevance of our approach? I give here three examples. One is, for example, in the longitudinal studies, where I can get samples at different times. I may not be able to analyze all the samples together, and so I will need to normalize different batches acquired at different times, for example. Another use of this type of normalization is during multi-center validation. And I will give you an example later. So this is the case where you analyze the same set of samples in two different places with two different setups to validate what you find in one cohort independently. And another useful application is when you have to acquire data with different platforms. And in this case, you would probably analyze samples with a triple quad or with a high resolution instrument from different vendors. And after this normalization, what you want is to be able to compare the data. And again, now I will give you a couple of examples of this. So in this case, we analyzed the human plasma from 22 different individuals. And to do that, we used different methods. In this case, the first method was a reverse phase MRM method, and the other one was a direct infusion high resolution MS. Different instruments. Of course, in this case, we use Agilent triple quadruple, and in this case, we used Thermo Q Exactive. Different operators that perform the experiments 1 year apart. Now, on this graph, we plotted the concentration values of 100 lipid species that we measured in these 22 individuals. On the y-axis, you see the concentration measured by reverse phase MRM, and on the x-axis, you see the concentration measured by high resolution direct infusion. As you can see from this data, the one on the upper part are represented in log scale, while the lower part are actually the actual values. You see that they are not correlating very well. This is before normalization. So what happened after we apply the same normalization I showed you before is that now the same data all lie on the line of equality here. So, and both, you can see both in the log scale, but also in the normal scale. And indeed, if then I represent the intermethod difference, like we did before, we see that pre-normalization, the RSD percentage is between 100 and 20%, while after applying the same normalization, the same data are now all below 20%. Another experiment that we performed, we increased the number of individuals analyzed, is 470 individual samples. And we also increased the number of lipids. In this case, we measured 250 lipids in each experiment. This case is of a multi-center validation. So we measure these samples here in Singapore, and then the same samples were measured in the lab of our collaborator, Peter Michael, at the Baker Institute in Melbourne. And in this case, instead of using a Nist reference material, we used just a shared pooled QC for external calibration. So both of us used the same QC, analyzed interspersed between study samples, and we used that to normalize all our data. The approach in terms of instrument was more similar compared to the one before. We both use reverse phase MRM detection, but it was done on two different triple quads, two different types of triple quads. Again, if we look at the data before normalization, we see that around the line of equality, there's still quite a bit of scattering of the data. Here we represented the concentration in Singapore versus the concentration in Melbourne. Now, again, what happened after normalization, you can see that now all the data for all the lipid species lie on the line of equality very well. And again, if we represent the percentage difference between the sites, in this case, before normalization is again between 80 and 20%, while after normalization is all data are well under 20% variability. So this is the idea that we had to improve harmonizing quantitative lipidomics data. In few words, you have to start by adding a reference material from the start of your analysis. If you think that using the Nist plasma sample is too expensive, or if you think that, for example, the Nist might run out of samples soon and you cannot use the same standard for normalization in the long term, there's still the alternative, as we shown in our data, to use your lab's long-term reference or even a full QC. What we suggest is maybe to then always keep a small amount of the SRM 1950 as a master reference, so that you can still recalibrate your internal reference material to that master reference, and then it will be easier to compare studies all over the world. And then you apply, as I showed you, a simple single point calibration to normalize your data. With this, I would like to thank the major contributor to this first part of my talk, not only Alex, but also Bopurla, Jaya, Jonga, and of course, Marcus Ben, who was the other mastermind behind all this project, and our collaborators here at NUHS, and of course, Peter Michael lab. Now, the second part of my talk is about how can we evolve from the approach that I just showed you. And one way is indeed to add this element here to our equation, which is the absolute concentration of each lipid in the reference material, so that we would be able to calculate quite well the concentration, the absolute concentration of the same lipid in our study sample. Sorry. And this is a requisite to generate those reference values that we want to have in the clinic. So to do that, we came up with the organization of a ring trial in which we want to measure lipid levels, absolute concentration of lipids in reference material, so that we will be able to generate biological reference ranges of lipids. Now, this is a collective effort together with many other contributors, in particular Lipid Maps, LSI, the International Lipidomics Society, and Avanti and Nist that supported this project from the very beginning. Now, to define a reference range in clinical chemistry, we have to go through different steps. First of all, of course, we have to identify the analyte. We have to measure it accurately and reproducibly. We have to define the populations for which the reference range will be calculated, and we have to agree on a statistical analysis approach. Now, what we thought of doing was to separate these different steps into two phases of our trial. So in phase one, we would only measure the analytes accurately and reproducibly, and then we will define the population in phase two. To give you just a little bit of historical view on how this started, all this initiative started from a meeting that Marcus Wenck and Andrei Shevchenko organized in Singapore in 2017, and where many of us met and discussed on how to proceed to be able to generate these reference values for lipids in human blood plasma. So what we did after that meeting were two things. One was to publish a position paper to develop accepted guidelines for lipidomics in human blood plasma from pre-analytics and post-analytics. And the other thing was to agree on how to better organize this ring trial. So what we came up with was that in phase one, we would distribute reference material and measure lipids absolute concentration in this reference material. By doing that, we would improve method standardization, but we would also establish a network of labs that then would work together also in phase two. And we decided to take a very focused approach and really concentrate on a few specific lipid species. And to start with, we decided to measure first ceramides and then follow up with bile acids, and then in the future focus on other lipid species. So as you can see from this slide now, the phase one for ceramides just ended. We collected all the results and we are now analyzing them. And I was lucky to be one of the coordinators of this study together with my colleagues, John Bowden, Kim Akros, Nils Hoffman, and Robert Harens. And now is already time to start with the phase one for the bile acids. And we will start soon sending around the samples for this new measurement. Now, why we decided to start with ceramides? And the reason is that for their use in the clinic. So ceramides have been associated with major adverse cardiovascular events, and plasma ceramides levels in blood can be used to identify cardiovascular risk through the use of several scoring systems. CERT1, I guess, is the most popular, but now it has been improved with CERT2 that includes additional lipid species, and there's also another one that is even more comprehensive, it's called SICK. Now, the idea of using ceramides to predict cardiovascular risk is one of the best examples on how to translate lipidomics measurement into the clinic. So the groups that started this, and I'm referring here to Zora Biosciences, but not only, started with an untargeted approach through shotgun and reverse phase MRM approaches, and a few hundreds of samples where they found that effectively this the level of few ceramides were changing significantly between healthy and individual at risk. In the end, they refined the list of analytes to 4 different ceramides that you see represented here, and they validated their targeted approach into bigger cohorts. And after that, this approach was adopted first by Mayo Clinic and more recently by Quest Diagnostics. So this is a very successful example on how starting with a high throughput lipidomics approach ended up with a real clinical test. So what we did, we shipped to 40 different labs around the world for samples. Nist was very supportive and shipped these samples for us, including the SRM 1950, of course, but also 3 new reference material that have been characterized by John Bowden's group in 2020. And then we ask the labs to measure only these 4 target ceramides. These are the ones that are used in the CERT1 score for the prediction of cardiovascular risk. And Avanti created for us a pre-mixed internal standard mix with the deuterated 4 ceramides premix at the right concentration to make things easier. In the end, we received back results from 34 different labs, and I have to say that this is a very good results considering that we ran this ring trial during COVID time. So I still thank all the participants for all these efforts. What we sent around together with the samples was also a validated SOP that was based on a published protocol. And this included extraction methods and analytical methods. We also sent to the participants a form to report the final results in terms of peak areas that we then use for blind data analysis. And the participants were also free to contribute measurement obtained not only with the methods that we suggested in the SOP, but also with their preferred analytical method. In the end, we got contributors sending, analyzing the samples with the standard method in 23 cases, and we had 15 participants that analyzed the sample with their preferred method. This preferred method included different type of extractions, different length of the run. Some were very short, some were very long, a flow injection approach, or even nano-SC. In terms of analyzers. Most of the participants used triple quads or Q-trap, 32 of them, but we also got results from people who used high-resolution mass spec, a few Q-tops and Orbitrap. Everyone managed to identify and quantify the 4 ceramides in all the samples. Now, just a few preliminary results to give you an idea of what we did. In this case, you see here the levels of the 4 different ceramides in the Nist SRM 1950. Each box represented in the graphs represents 1 lab, so these numbers are all the different labs. And these are the levels of the ceramides. So just a few words about these first preliminary results. Here in brown, you see results from the same lab that repeated the measurement after storing the extract for quite a few days. This was relevant for us because to some country we were not allowed to ship a plasma sample, so we had to extract the samples here in Singapore and ship the extracts. And this allowed us to understand that the extract was stable enough. And indeed, here, we don't know which ones, but the labs that received the extract perform comparably with the labs that receive the fresh samples. And another observation is that when using preferred methods instead of the standard method that we suggested, the concentration value aligned with all the other labs. You see here in blue, 2 of these labs decided to use that preferred method, and indeed it perfectly aligns with the standard method. And in green, of course, you see that there is also the presence of outliers. These outliers were different. In some cases, they measured completely different levels for all the 4 analytes, but in other cases, only 2 analytes were really off and others 2 were aligned with the other labs. And in other cases, even only 1 analyte was not aligned. Some of these disparities were lessened after we applied the calibration curves that we asked to build with the experiments that were run. In other cases, the correction didn't really help, and we are trying to understand as part of the project what are the issues there, if the ROM peak was integrated or if there are other reasons. But this will also be very interesting because it will allow us to improve our methodologies. This figure instead basically answered the main question that all the participants have, which is, are we all measuring similar values? So what we represented here is the CV percentage between different labs for the 4 different ceramides in case the measurement was calculated after single point calibration or after using a calibration curve. Each of these little square is a different lab, and you see the distribution of the CV percentage. So in the end, what we can say is that the reproducibility is quite good. After applying single point calibration and eliminating just a few outliers, we obtain CV values between 11 and 15%. So the results were quite reproducible, and even after using the calibration curve, the CV percentage was between 18 and 20%. So in the end, I think we all measure very similar values. Now we are ready for phase 2 of this trial, which means we will want to measure reference ranges in human population, both healthy or not. What will be very important in this phase is to use what we learned from phase 1, to use the network of labs that we created. So different labs will probably have access to different human cohorts, so that will generate reference values for in very different conditions and in very different human cohorts. And the other important aspect will be the engagement with the medical community, because they know better how to define the study populations, and they are the ones that have access to these sample cohorts that we can measure. So I also hope that some of the attendees of this talk will be interested in joining phase 2. So please get in contact with us if you are interested in this exercise. With this, I would like to end both my presentations. I still want to thank all the participants of this big study, and all the other contributors, the International Lipidome Society, Nist, and Avanti in particular. And I would like to thank you for attending this talk, and I will be glad to answer any question you have. Thank you.
All right, thank you so much, Federico. That was a really interesting talk. One question that relates to what you were talking about at the end that came in is from Dipali Kale, and it is, what is the procedure to be a part of these trials?
Yeah, thank you. Thanks for the interest. So I think you might have my email. It was in the last slide, but it's also easy to find on the internet in our website. So please get in contact with me or any of us of the Lipidome Society, and we will explain how to participate to the phase 2.
Okay, great. A question that came to my mind when talking about moving lipidomics into the clinic was about normalization. And I was curious about the variability in the data following normalization. How does that compare to other clinical chemistry data?
Yeah.
For things that have moved into the clinic, I guess.
Yeah, so I'm not an expert of clinical chemistry, of course, but we are all aware that they have to go under strict validation, right? They have reference samples that they share in the community, and that they have to measure periodically, and they have to show that they are measuring all the same values. So definitely, so far, at least, and that's what we saw from that first paper in 2017, our variability is not comparable to that. But I think that the community is working towards getting to those reproducibility values, and we are doing, I explain how we are doing this, but there is also a clinical lipidomics group inside the International Lipidome Society that is now thinking of running another ring trial to exactly fix those issues. But I think that what we are showing now is that, you know, when we use the right normalization, and when we have the right standards, of course, I think we can be also very reproducible.
Yeah. Great. Another question that came in is, which extraction solvent do you find most suitable for standardizing lipidomic sample preparations?
Yeah, that's also a good question, because all of us, I think, use slightly different protocols. But I think that over the years, we've shown that there are many different extraction protocols that are very reproducible, and that can be two-phase extraction protocols that are historically the most used, and I'm referring here to Bly and Dyer on forge methods. But also single-phase extraction. We have a lot of experience with butanol-methanol single-phase extraction that is simpler, and it works pretty well in terms of reproducibility. If we really want to go into the details, maybe we should clarify that there are methods that might work better for specific classes. So there are methods that have been shown that they are less reproducible for maybe one class or two classes when you try to do a comprehensive analysis. So in that case, maybe depending on what you want to analyze, there are a lot of data and literature that clarify that.
Yeah. For high throughput, would you say that single phase may be the way to go just because of the ability to automate?
Yeah, exactly. So we thought about that when we had to automate it. I mean, of course, even when you perform it manually, a single phase extraction is much easier to do and faster. But even with the robotic system, unless you have the most advanced one, it's easier for a simpler robot to do a one-phase extraction, and that's what we did exactly. So now, because you don't have to probe the height of the lower phase, and you have less chances of contamination. So we also optimize automated extraction with a robotic system in a single phase way. Yeah, you're right.
Great. Another question about the reference materials. Do you know if they're isolated from one individual only, or if they're a pool? Would they, for example, would they serve equally for males and for females?
Yeah, so they are usually pooled because, of course, they are more representative of the metrics that you are studying. For example, in the case of the SRM 1950, I think it was 100 individuals from different ethnicities in the US, and of course, different sex representation. So that's quite a representative sample, and that's why also is one of our favorite reference materials. For the last used one, maybe they might have used a smaller number of individuals, but they are usually always pooled samples. Yeah.
Great. In moving lipidomics toward the clinic, do you think that there's room for untargeted lipidomics, or do you think that the focus needs to be shifted towards targeted, like in the ceramide ring trials and the bile acid trials, where you're measuring a predefined list of lipids?
Yeah, also good question. I hope that there is room for untargeted, and what I think is that probably we need different phases, probably because of the analysis in the clinic nowadays are done. It will be easier to enter with a targeted method in which we measure a smaller number of analytes. But our community, and again, the clinical lipidomics group in the Lipidomics Society especially, is working towards more comprehensive approaches and trying to find out which is the best way to have some sort of untargeted approach. Also because in a way we would use better all the potential that lipidomics has, and because there's also limitation in the number of standards that we can use to quantify these species. So of course, one of the reasons why we chose the 4 ceramides is that Avanti has the for the iterated standard, so it makes quantification very easy. But we are now working in combining maybe a few standards per class and see what is the best mix to get to a good quantification. So we are working, for example, on using your ultimate splash mix to understand what is the best way to use it. But of course, this is not so trivial. So I guess that we will still need some time to get there.
Yeah. I believe we may have reached the end of our audience questions. Oh, here we go. A follow-up question. Could pooling samples into a reference material disguise results for a specific ethnicity?
Sorry, what is the question about the ethnicity?
So if you're pooling samples, could that maybe mask differences between ethnicities if you have a multi-ethnicity pool?
Oh, yeah. I mean, so we have to distinguish between the use of reference sample for normalization and what we want to look at in the studies. So we all know that when we do cohort studies, we have to stratify our batches or our sample analysis using all these criteria that could be, again, sex, ethnicity, or other factors. So all those other factors will have to be considered during analysis. But then the way we use the reference material is different. So that will not be so specific, but it will only be used to then normalize our quantitative measurement. So in that case, it's not so important.
Okay. And so that's part of the phase 2 of these trials, in a sense, if I'm correct. Phase 1 is establishing the general reference ranges and then going into the individual populations and looking for variability. Is that correct?
Yeah, absolutely right. I mean, phase 1 was, okay, how can we get the right methods? How can we create a network that has the right knowledge to perform this study based on our experience, based on our mistakes, based on what we learned from phase 1, right? And then all these labs and even more, hopefully, will have access to different cohorts. But now we will know how to properly measure these ceramides in all these different cohorts. And these different cohorts, as the previous question was pointing out, will for sure include different ethnicities, and that will be representative of different diseases. So they will have their internal control, but by having a shared reference material between all these groups that we decide before starting phase 2, we will then have a common calibrant for our external calibration. So all these studies made on different sets of samples, made in different ways on different instruments, if people don't want to use the standard approach, will still be comparable.
Yeah, exactly. Yeah, a question that came in sort of regards the cost of Nist plasma. Avanti does not offer Nist plasma. That comes from Nist or other vendors, but it is it can be expensive. So could you talk a little bit more about the alternatives that you mentioned in your talk to using Nist plasma?
Yeah, sure. Yes, this is a very good point. We thought about that, and it's true that can be a bit expensive. Although, of course, you don't use so much of that plasma during your analysis, because what we suggested was to include one aliquot in each batch that you measure. But again, yes, it can be expensive. So what we show is that you can use any other shared material between the different studies for normalization. So it can be something you already have in your freezer in a decent amount, of course, because you just have to consider that you will want to use it in all your future experiments. Or in case you want to compare your results with another lab, you will have to share this material. But again, it could be any material of the same matrix. So again, of course, we are talking about plasma. So you can share your plasma as we did with our collaborators in Melbourne, and then use that one to normalize. What I was also suggesting is that for future comparison with published studies that might have used the 9050 from the Nist, if you still keep some Nist in your lab, you can recalibrate your own reference material to the Nist one, right? And so being able to then renormalize all your results to the Nist in the very end. So you don't need to run the Nist plasma in all your batches. You will run your own reference material. But if you run once in a while the Nist, you will be able to recalibrate your reference material and then, as a consequence, recalibrate all your study samples, and that will increase the comparability of your results. I hope I was clear with this, but if I wasn't, yeah. So, but it's a very good point, and there's, we even put it in the discussion in our paper because we realized that it might not be accessible to everyone, or, even if there's a lot of this material, one might think, oh, maybe there will not be enough in the future, while in my freezer I have 2 liters of my favorite plasma and I want to use that. So that makes total sense.
Yeah, absolutely. Nist SRM 1950 can't last forever.
Yeah.
All right. With that, I think we're out of questions. So thank you so much, Federico, for doing this. We really appreciate it. And thank you to everyone for joining us this morning or this evening. And we'll see you all at our third installment of Explore the Lipidome. And again, thank you very much, Federico, for joining us today.
Thank you. Thank you for having me.
Of course. Have a nice evening.
You too. Bye, everyone.